Hello, everyone, and thank you for joining us today. It's wonderful to see so many families, clinicians, researchers, and advocates joining us once again from all around the world. Thank you for being part of this growing global community. One of the most encouraging aspects of neuroblastoma research is that progress doesn't happen in isolation. It happens because experts across countries and continents work together, share knowledge, and build clinical trials that help answer some of the most important questions in pediatric cancer. Today, we're honored to welcome Dr. Lucas Moreno, president of SIOPEN. SIOPEN is one of the world's largest collaborative neuroblastoma research network, bringing together physicians and researchers across Europe to develop clinical trials, establish treatment standards, and accelerate discoveries that benefits children worldwide. Through close collaboration with international partners, including colleagues in North America and beyond, SIOPEN continues to play a vital role in advancing the field. Today, Dr. Moreno will share updates from SIOPEN, including the latest developments in clinical trials, research initiatives, and the future direction of neuroblastoma care in Europe. For families, these updates provide more than just information. They offer a better understanding of where the field is heading and the progress being made through global collaborations. Please feel free to ask questions in the chat below. Dr. Moreno, thank you for joining us today. We truly appreciate your leadership and your commitment to improving outcomes for children with neuroblastoma, and we look forward to learning from you.
Thank you very much, Einat. It's a pleasure to be here giving an update on what we're doing from SIOPEN. So hi everyone. I'll be sharing my slides in a second, and I hope we can start it soon. I hope you can all see the slides now that it's that it's working. So yes, I'll be giving an update on SIOPEN. I'm Lucas Moreno, pediatric oncologist. I work in Valdebrón Hospital in Barcelona in Spain, and have dedicated my career to neuroblastoma and drug development. And I'm the current president of SIOPEN, which is the cooperative group that started in Europe, but now it's growing and has a global perspective as well. So here are my conflicts of interest. So I'll try to cover during today a number of areas of the neuroblastoma work that we're doing. An update from SIOPEN, part of what we're doing for high-risk neuroblastoma, for non-high-risk neuroblastoma, for patients with relapsed disease, and some other initiatives and projects. Some of it some of the perspective or the background I already shared during last year, but hopefully there's there has been a number of projects and news and aspects that have changed this year, so it's really worth sharing this update. First, just to say from SIOPEN, we, as I said, we have got more than 600 members from 27 countries. We started in Europe, but now we also have members we have countries as members with Israel, Australia, New Zealand, and then also members from different parts from other parts of the world. We changed the structure of the organization about 2-3 years ago. Now I'm really honored to work with 2 vice presidents and a manager, and very closely with patient advocates as well that help us incorporating and working with patient advocates from the beginning of all initiatives. We have got 3 main focuses on what we do: translational research, clinical trial development and delivery, and data management aspects which we work on. And I think we start with this point where we are very much aware that still there are many children with high-risk neuroblastoma that develop relapse and have poor outcomes, and that's something that we need to change and continue changing. We cannot accept the results of current therapy. We are witnessing major advances in basic and translational research. We are seeing promising results from early phase trials and new drugs, but still we have many challenges to take these innovations into the front line. We still have significant challenges to run the clinical trials that we need to take these drugs into the front line. We also have many challenges and disparities to introduce these new drugs into routine care. And what is true is that international collaboration and multi-stakeholder collaboration is increasing, and this we are sure that will facilitate a better future. This is a brief summary of the current high-risk neuroblastoma therapy, just as an introduction. We have got induction chemotherapy, for which we use the rapid Kojic chemotherapy regimen that we have been using for the last couple of decades, which is chemotherapy given every 10 days for 80 days. We then do the stem cell collection surgery of the primary tumor, to then follow into consolidation with high-dose chemotherapy with stem cell autologous stem cell rescue, and we use busulfan melphalan. We then administer radiation therapy to the primary tumor, and then in the maintenance or post-consolidation therapy, we give anti-GD2 therapy with dinutuximab beta in a long-term infusion. And this is based on the experience from the previous trial that was run in the 2000s. But still, despite the improvements with immunotherapy, more than half of the children experience relapse. So we do need to continue improving this, and we do this through clinical trials. This is just to illustrate how treatment fails. There are a group of patients that have got refractory disease, and this is after induction. They don't have a sufficiently good response to treatment. And then there are patients whose disease progresses or relapses during treatment and during follow-up. And all of these groups of patients need better treatment outcomes. The current trial for neuroblastoma in SIOPEN is the high-risk neuroblastoma 2 trial, which is currently open in all the countries. At last, it's open in all the countries that was initially planned. It opened in France in 2019, and it's now open across multiple countries in Europe, Israel, and Australia. And the trial has got a rapid Kojic induction. We started with a randomization in induction with the German GPOH regimen, but that had to close because of problems of supply with one of the drugs in the German group, in the German regimen. So the patients receive the rapid Kojic induction. If they have got an adequate response, they continue with surgery. They are offered a randomization between 1 versus 2 transplants, and this is based on the results from North America that showed that 2 transplants had better results than 1. And then we follow to radiotherapy with consolidation of the dose or the way that we give radiotherapy, and then everyone gets immunotherapy with dinutuximab beta. The patients that have insufficient response have chemoimmunotherapy within the trial, with chemotherapy with dinutuximab beta, and then they follow to tandem transplant and radiotherapy and immunotherapy. That's the standard, the frontline trial. We have recruited more than 800 patients, but still we want to do amendments in this trial to improve and ask other questions. We are piloting the combination of chemotherapy, of the rapid Kojic chemotherapy, which is here below, given every 10 days with all these courses, together with the anti-GD2 therapy with dinutuximab beta, and also with the German regimen, and that's why we're doing this SIOPEN pilot trial. This trial is already open in the Netherlands, France, Italy, and Spain, and will open soon in 2 or 3 more countries. We have already completed the first cohort of rapid Kojic with dinutuximab beta. We are now recruiting an expansion cohort, so this is a message for newly diagnosed, to be aware that patients with newly diagnosed high-risk neuroblastoma in these countries can benefit from participating on this trial, which is where all patients get chemoimmunotherapy, and then in a few months we will do an amendment to also test the GPOH regimen, mainly to be conducted in Germany, where that regimen is still used. Still, as I mentioned, we want to ask more questions in the high-risk 2 trial. The first one that we have been pursuing for a very long time is to evaluate the role of ALK inhibitor therapy with lorlatinib for patients that have ALK genomic aberrations, so mutations or amplifications of ALK. And this is a very long endeavor where we have been trying to start this modification of the treatment. We have had numerous challenges to get supply of the drug, to get funding for the trial, and to get regulatory advice and approval, to be sure that the trial will help to have access to lorlatinib in the future if the trial is successful. And now we are in the final preparations to submit this amendment after summer. So hopefully, we expect that by the end of the year, this will be already available, but it's been already a very long time since we wanted to start this, and we haven't managed to open it yet. We are already giving chemoimmunotherapy for patients that don't have that have an insufficient response, and we want to expand that so that if the trial continues for 3-4 more years, all the patients that have insufficient response have access to chemoimmunotherapy, and this will be also very valuable data for this group of patients. We also want to evaluate chemoimmunotherapy upfront. So the results of the data that we will get from the pilot study that I mentioned of rapid Kojek with dinutuximab beta, there will be a randomization introduced in the trial also we're working on it for next year, so that we give to some patients chemotherapy and to others chemotherapy with immunotherapy upfront. We are having some discussions about the use of DFMO, but this is only preliminary, and we're really not sure if this will take place now that the company that was developing DFMO in Europe has withdrawn their expanded access program. And we have already started discussions for the high-risk 3 trial, willing to work towards a new international induction regimen so that we use the same regimen in both sides of the Atlantic to evaluate new consolidation ideas, hopefully ideally with CAR T cell therapy, and to pilot and implement improvements of anti-GD2 in the post-consolidation setting. A brief mention of the LINES trial, which is our low and intermediate risk European study that closed some years ago, and now we're sharing the results. There we have shown some data in the past ANR. We will be showing some more data in SIOP this year. Patients that are not high risk are in the low risk or intermediate risk categories, and there are some patients with neonatal adrenal masses. So this is just to summarize that we have found out with this trial that for low risk patients, adapting treatment to their genomic profile, metastatic burden, and symptoms allowed to give no or minimal treatment with excellent results. For the intermediate risk patients, we still need more work to identify the best genomic markers, because there are still patients that don't have so good results and need probably more intensive or more innovative therapy in some patient groups. So the main theme in the non-high risk patients is that using better markers will help us to better stratify these patients and say who has higher or lower risk of relapse, and then we can give the minimal therapy needed for each of the groups. And we will now move to the relapse setting, where there are many options, and we're aware that often patients and families have to navigate through a wealth of options and possibilities, and it's difficult to understand why are there so many options. I'll try to put them in order. First, this slide tries to summarize what we think and hope is the treatment for patients with first relapse. So once neuroblastoma has appeared again for the first time, we know that this is not good news, and that we still need to improve frontline therapy to avoid as many relapses as possible. But since they are still occurring, we want to recommend a strategy that is similar to the initial one, in that it has different phases, like a reinduction and then consolidation and maintenance or post-consolidation options. In patients with first relapse neuroblastoma, this year we launched the SIOPEN guidelines for first relapse neuroblastoma, where we recommend that every patient should be attempted to have a molecular profile of the tumor biopsied at relapse, because their tumors may have different genomic aberrations at the time of relapse. We are setting up and opening across Europe and the network the Beacon-2 trial, and then the options include, and I'll talk about the trial a bit later. The options for patients include chemoimmunotherapy, the RIST regimen that has been shown by the German group, particularly for patients with MECAN amplification, bevacizumab with irinotecan and temozolomide chemotherapy, and some targeted therapies in case of an ALK aberration, for example. The patients could get chemotherapy with an ALK inhibitor. In regards to consolidation, patients can receive local control if there is an isolated metastasis or primary tumor. They can receive radiotherapy or surgery. There are consolidation options like the CAR-T trials. And there are some of them in Rome, in Berlin, in London being developed in the network. The German group has used the haploidentical transplant in many patients. There are radionuclides like MABG therapy, the Minivan trial that is now closed, the Ludo-N trial. And then for maintenance, we need some low toxicity drugs. And there's not a lot of data still on vaccines, on DFMO, but there's different options, including targeted therapies, continuation of the reinduction chemotherapy, more oral or metronomic chemotherapies, or participation into early phase trials. But we think that there should be this planning of the whole treatment for the first relapse, trying to tackle all these aspects. This is not new data, but this is just to summarize that we conducted the Beacon-1 and the Beacon Immuno data some years ago, and that we presented the results, and we showed good data for two combinations. One is bevacizumab, irinotecan, temozolomide, that showed different data than all the other arms, and then dinutuximab beta with topotecan and temozolomide. This is chemoimmunotherapy following the results of the COG ANBL 1221 trial. So with these data is with what we used to plan the next Beacon 2 trial that I'll mention in a second. But I wanted to touch base on some of the data that is emerging from the Beacon 1 trial and that Gudrun Schleiermacher presented recently at AACR. And this is just to illustrate how the samples that we collect during the trials also provide very useful value and very interesting type of data. We collected data from the Beacon biology study from close to 200 patients from many of the participating countries. And we are also, with data sharing initiatives, we are also putting together data from more patients, because if these patients had their tumor sequenced as part of their national tumor sequencing program in France or in the UK, in other countries, we are able to retrieve that data and enlarge the data sets. And we collected data and samples from the patient's tumors, but also from blood samples at the beginning of treatment, and then after 2 cycles, after 4 cycles, after 6, and then at the end of treatment. And this work led by Gudrun Schleiermacher and Sally George in Institut Curie in France and Institute of Cancer Research in London. They have done whole exome sequencing, panels of gene panels in the tumors, and then sequencing of the circulating free DNA. As well on the different plasma samples, which, for example, in this graph, we show all the different alterations that each of the patients has. Each of these numbers in the bottom is one of the patients, and they have different chromosomal or genomic aberrations. Some of them are more frequent, like, of course, MECAN amplification or alterations of the RAS-MAPK genes. But others are less frequent, and we now have a clear perspective of the landscape of neuroblastoma tumors at the time of diagnosis, but also at the time of relapse. But interestingly, also we also have data on the sequential samples, and here we can see we also measured the amount of circulating DNA in the plasma samples in the blood of the patients when they entered the study, and we showed that those patients that had a higher content of tumor had worse outcomes, and that we could measure this content. And here you can see in the different time points. So in red, when they started on the Beacon trial, after 2 cycles, after 4, we see how the overall DNA content comes down, and then it comes back up in some patients if they experience relapse at the end of treatment. And we see that how this changes in different patients. But we are also seeing that the same patients have different clones, so different proportions of mutations or different mutations in different parts of the tumor. And we are seeing this in this graph at different points in time, when they're on treatment, when they respond. And this help us to understand the clonal evolution of the tumor. And there is a lot of work trying to understand what happens to the tumors when they are under the pressure of the treatment, that is particularly when they treat with the chemotherapy, the chemoimmunotherapy is working, how the tumors learn to escape. So there's a lot of data coming out of this, and which is quite novel, because we have got a substantial number of patients together with a very rich clinical database, and it's to put in the value, to show the value of the samples that we collect, because we learn from the neuroblastoma biology, and this help us to design better the treatments of the future. We will now move to the Beacon 2 trial that we are also rolling out in the different countries. This trial has the main randomization is for patients with first relapsed neuroblastoma. They are randomized to the two winners of the previous Beacon trial. One is chemoimmunotherapy, the other is bevacizumab, irinotecan, and temozolomide. This is the randomization at the moment, but we hope that we will have more arms to add to this randomization so that we can test more novel approaches in the same trial. There is also what we call a tier 2, which is for subsequent relapses. So patients in second or third or fourth relapse can receive more innovative arms. And for example, we have already completed this arm C, which is the chemotherapy with the dinutuximab beta and with the bevacizumab. So altogether, we have already treated patients in this arm C. It is completed, and we're now waiting to analyze the result. And our aim is to include new arms, arm D, arm E, etc., in this tier 2. Once we show these new combinations are safe, then we will move them into a new randomization arm so that we speed up the development of these trials. We don't have to set up a whole new trial for each of the arms. We use what we call a platform trial. So the current tier 1, as I mentioned, this big randomization is for patients with first relapsed high-risk neuroblastoma. The second is for the tier 2 is for subsequent relapses for patients older than 1 year with adequate organ function. The trial is now open in the UK, in Denmark, and Australia, and we're hoping to open soon in the rest of the European Union countries that are already part of the trial, Israel and New Zealand. We have already randomized 18 patients in tier 1, and the tier 2 arm C is completed, and we're preparing more arms. We have the challenges of working with different pharmaceutical companies for them to provide the agent for the different combinations, but we're now closer to securing drug supply for 2 more arms, and we're hoping to open them soon. And we're also working to make this fit for regulatory standards, because it is important that the companies perceive that the Beacon trial is a good way to fulfill their regulatory obligations. So we have already consulted the European Medicines Agency for tier 1, and we're preparing a consultation for tier 2 so that the different pharmaceutical companies can be ensured, can understand that if the drugs are tested in the Beacon trial, in the Beacon 2, they will fulfill the regulatory obligations. And we think this will help to have more arms and speed up the development. Here are some examples of agents that we are trying to have as potential future arms. Some of them we have discussed with the companies, but they have discontinued the development of these agents, like anti-CD47, ISRH2 inhibitors. Some others, like the copper chelator, trientine, or 9-ING41, are closer, and the companies are willing to work with us, and we hope that these two will be the next ones. Some others will be developed in other cooperative groups. Some others we continue to generate some more preclinical data, and something that we've also advanced is that there has been a funding call by Solving Kids Cancer to fund preclinical data that facilitates arms for Beacon 2. So that's soon to start, so that there is all the data that is necessary is generated, and we have more options in the trial. So um, beyond the first relapse, where we recommend that all patients enter in the Beacon 2 trial, what other data is available, and what are the other therapies are out there? I won't mention in detail the CAR T GD2, because I know it's been the matter of other talks, but just to highlight that the Rome team, the team from Bambino Gesù, has been developing a phase 2 trial in the multi-center setting, and now this is finally advancing to happen, and they are working on the preparation, on the regulatory steps needed for this preparation. So we're really hoping that soon, in few months' time, this trial will be open, not just in Rome, but it will be open in some other European countries. I mentioned to the radionuclide trials. These are, in general, useful part for as an option for consolidation. We have completed the Minivan trial that tests MIBG therapy with nivolumab, an anti-PD-1 agent, and with dinutuximab beta. We have shown that it's safe and feasible. We had a nice response rate of 43% in the cohort 3, the one that received the full dose and was expanded. And we're hoping to have the full analysis of all the patients and the detailed data at the end of this year. And there's also a trial of lutetium dotatate run by Jakob Stenman from Karolinska in collaboration with other centers in Europe, that has recruited the first stage with the first few patients and is now recruiting the second stage. And they are also evaluating other combinations and radionuclides, when we think there's a whole space of new agents in this space. Here, I also wanted to touch base or mention that for patients with subsequent relapses, ITCC and SIOPEN work on a portfolio of early phase trials. This is difficult to list at one single point because many of these trials open and close often, but it's really advisable to consult with the large trial centers in case of a need for a clinical trial. There are biomarker-driven trials for patients with ALK-positive disease, but also for FGFR mutations, for RAF alterations. There are different radionuclide trials, with the LUDO-N being open, as I mentioned, and there's another one called Int'l that is open in some European centers as well. There are targeted agents for different combinations. So the Nivent trial is a combination of an anti-PD-1 with an HDAC inhibitor. ABTL is another dual PI3K and autophagy inhibitor. There is a B7H3 ADC trial opening. There are different arms of the eSMART trial that is open in ITCC, and the patients receive different treatments depending on their alteration, depending on their molecular profile. There's another option of a targeted agent with a radionuclide being developed. And with the neuroblastoma focus, there's still the naxitamab phase 2 trial is close to being completed, but there's in setup a trial for relapse and refractory neuroblastoma with the St. Jude antibody. There are other CAR T trials. There's a trial in setup with tumor infiltrating lymphocytes. So we still would like to have more trials available for patients with neuroblastoma. We would still want that more patients with neuroblastoma benefit from these trials, because they remain available in a relatively small group of centers. Probably 10 or 15 hospitals in Europe have access to most trials, but this does not provide good enough coverage. So in summary, of the advances we are seeing with the clinical trials, I think I wanted to highlight that every time that I give a talk, I try to reflect: are we really making progress? And just highlight the changes this year. So on the high-risk neuroblastoma 2 trial that is ongoing, it's now open in all planned countries. It was not open in 6 countries that were waiting for some time to be open, and now it's open. There have been a lot of discussions with the European regulators to make sure that the results of the new arms that we want to have are valid for the future, so that after the trial, if the trial is successful, the patients in the future will be able to get access to the drugs. And at the moment, we are preparing these lorlatinib and dinutuximab beta amendments. The Beacon 2 is finally open internationally, and it also has got this regulatory validation, and the data can be used for regulatory purposes. And we have completed the ARM-C and are working on ARMS-D and E. We are also working internationally with our colleagues in the Children's Oncology Group to try to develop a pilot study of a new induction, because we know that both inductions, the one that is used in the Children's Oncology Group and in SIOPEN, both have limitations. Still, not all the patients achieve a complete response, even with the addition of chemoimmunotherapy. We think that there will be a need for improvements. So we are trying to converge and develop a regimen that could be used in the two in it across the globe, and are having discussions with pharma. This is a bit still far from happening, but I think it's very inspiring for us to be working on trying to join forces with internationally. With Minivan, we have advanced on all the work to analyze the data, and that will be ready by the end of this year. And with the CAR-T GD2 also, that the phase 2 has taken quite a long time on the regulatory validation, but it is advancing and moving forward to be available soon. I also wanted to touch base on a couple of other SIOPEN international initiatives, like clinical practice guidelines that we have launched within SIOPEN for patients with that have the first relapse of neuroblastoma, that have relapses in the CNS, in the central nervous system, that have relapses with ALK positive mutation, with ALK mutations or aberrations, with FGFR. These are available for SIOPEN members, and we hope they will improve to the treatment of patients across the whole network. We also have high risk and low intermediate risk guidelines that we're hoping to update during this coming year, and are developing other consensus documents on improving how we use immunotherapy. We have got the SIOPEN bioportal, which is a database of all the SIOPEN patients, that works like a database with a virtual biobank to facilitate the research and understanding that what happens to patients. They go into this trial, then they continue on this other trial. They have a relapse and they go on another trial, so that we can understand and track the patient pathway for all the patients with neuroblastoma in the network. It has started recruitment with more than 150 patients recruited, and 9 countries already open and others opening. And this is linked to the genomic databases with EUPID, which is a European patient anonymization system to allow understanding the whole pathway of the patients. We have also started the Mona Lisa trial, which is a pragmatic clinical trial to understand the role of liquid biopsy in patients with relapsed neuroblastoma, where the patients are randomized to have liquid biopsy performed in real time, or have the results retrospectively reviewed. And we have already started recruitment for this trial in Princess Maxima in Utrecht, in the Netherlands, and we are linking this, expanding this trial, and hoping that this will really make it a clinically valid decision-making tool in the future. Also, to mention a couple of other multi-stakeholder initiatives that try to merge, to put together all the stakeholders. The neuroblastoma drug development strategy are groups of clinicians and biologists, mainly with some input from regulators. We have had 3 already. We're having the 4th one in Utrecht this Thursday. So we are prioritizing different targets and drugs in different areas. And something that we continue to work is to improve the supply of drugs from pharma for the clinical trials. There's an aspect of having more availability to treat the patients in clinical care, but also to have more collaboration from the different pharmaceutical companies to speed up some of the trials. And also Accelerate, that puts together the regulators, pharmaceutical companies, patient advocates, and the clinicians. And there was a couple of years ago, there was a pediatric strategy forum on anti-GD2 therapies. This year, there is one strategy forum on B7H3, another target of high interest for neuroblastoma. And wrapping up now, SIOPEN has, I wanted to give this highlight on the geographical perspective. We have a growing global community. We are strengthening our collaboration, not just within the SIOPEN network, but with new countries like Turkey, Middle East, Asia, South America, with more people, clinicians interested to join to join the network. We are really committed to harmonize and improve frontline therapy. We need to improve our operational capacity to expand the clinical trial networks to all these new areas. In terms of cross-border collaboration, I think there is a very positive aspect that is we really work very closely with the Children's Oncology Group, with ITCC, the European Early Phase Trial Consortium, and with other groups. And something that we are still working together with ITCC and with patient advocacy groups is to facilitate the cross-border trial participation. This is really limited. There are only some successful examples in the Nordic countries, but in general, it is very difficult to send patients from one country to the other. And this is really putting a limitation to many patients and families. And we know that there are some successful examples and want to capitalize on those. And finally, in terms of our SIOPEN advocacy group, the group is growing and is active, but we really share the aim of having representatives in all the countries, not just a group of people that is interested in neuroblastoma, but try to expand the network. And we're really happy if you want to get in touch with our SIOPEN advocacy group. We are also working on we drew our agenda for the future together with colleagues in North America and with patient advocates that we need. And we're working on the action points to deliver. Some of them are harder than others, of course, but we really want to move towards this international strategy for trials, for bringing new drugs into frontline, to facilitate access to effective therapies, to have less toxic therapies, and to improve this access across the globe and making it easier for the patients and families. So to wrap up, I'll summarize saying that we are very much aware that many children with high-risk neuroblastoma develop relapse and have poor outcomes. There have been major advances in research and in early phase clinical trials, but we still have these substantial challenges to conduct frontline and early phase trials and introduce new drugs into routine care. But we are definitely working on it, and we see the advances day by day, and hope that all of this together will help us to improve the landscape. So thank you very much. I'm really happy, really thankful to ANET for inviting me to give this summary. And I'm really happy to take any questions. I see there is a question about tandem transplant. So patients that have a good response in the high-risk 2 neuroblastoma trials are offered the randomization. So in Europe, still the standard therapy is one transplant with busulfan melphalan. So if they have an adequate response, then the randomization of 1 versus 2 transplant is offered. That is the main randomization of the trial at the moment. And then patients that have an insufficient response, then straight away are offered the 2 transplants, because there is data supporting their use for patients with insufficient response. We don't have partnership with colleagues in Brazil, but we're really happy to try to have them. I mean, indeed, I'm traveling to Brazil in 3 months' time, and I hope I'll be able to strengthen that aspect. There is another question about, besides DFMO and the vaccine offered an MSK, are there other routine treatments to prevent relapses? There is a bit of a semantic aspect in this question, that all the treatments are given to prevent relapses. So yes, there are more treatments that can be given. There is no prevention mechanism that DFMO or the vaccine have a way of prevent relapses that the other therapies don't have. all the treatments that we give after the patient has responded are given to prevent a response. So the patients there are patients that use oral chemotherapy regimens, metronomic regimens, that receive a targeted therapy. There are different types of treatments that can be offered at once the patient has finalized the treatment for relapse. None of them are perfect, unfortunately, because still many patients relapse during the treatment during the maintenance treatment.
Okay.
Mhm.
Thank you so much, Dr. Moreno, for your time, your insights, your continued leadership. We really appreciate it. Your work through SIOPEN and beyond obviously demonstrates the power of international collaboration and reminds us that meaningful progress comes from researchers and clinicians working together across borders. To everyone join us today, thank you. Your curiosity, engagement, and commitment to learning continue what makes this community stronger. Every question asked, every session attended, bring us one step closer to a better future for our children. As we look ahead, our next webinar will take place in August 5th, where we'll be honored to welcome Dr. Francesca Del Buffalo for an exciting session on CAR T cell therapy. Dr. Buffalo will share updates on one of the most promising areas in neuroblastoma, as Dr. Moreno just mentioned, helping us better understand how this innovation approach is evolving and what it may mean for our children and families in the future. We hope you'll continue to join us as we come together, leading experts from around the world to share the latest advances in research, clinical care, and innovation. Until next time, take care, stay informed, stay connected, and remember, together we can change the future of children with neuroblastoma. Thank you.